Welcome to the Tree of Life podcast. My name is Joel Ledford, and I have the privilege today of being with Dr. Mitchell Singer. Mitch Singer, welcome.
Thank you. This is great.
So one of the first things that we do in these interviews is we ask you about your background in science. And so I'm really interested to know, did you end up at UC Davis? What was the path? What got you interested in science?
So I have to say, what really got me interested in science was being a complete and utter science nerd in high school. You know, science club, all that. My father was a microbiologist, so he got me really early on looking at microbes. I remember going to his lab as a little kid and getting all excited. So I have to say, he's the real reason I went into microbiology. I then went to the University of Illinois and worked with Sam Kaplan on photosynthetic bacteria, which was like my dream come true because my science project in high schools were about photosynthetic bacteria. So working with Sam Kaplan was literally like working with a rock star as far as I was concerned. So I was so psyched.
Wow.
Really nerdy, I know. Then I went to Madison, Wisconsin to get my PhD in bacteriology. And the one advice Sam Kaplan told me was, there's a lot of interesting microbes, don't work on E. coli. What did I work on? E. coli. Okay, so I worked for Carol Gross, and I was trained as a classical geneticist, molecular biologist. I learned so much. She was an amazing mentor. And from there, I went to Stanford University to work with Dale Kaiser on myxobacteria. So E. coli is a phenomenal organism to learn on, but it doesn't do a whole lot. I mean, now there's all the work on pathogenesis it does, but for the most part, it's really limited in what it can do. And I wanted to take those skills and apply it to something novel in the microbial world, and that was myxobacteria, because here are single-celled microbes that undergo a developmental program and form these multicellular fruiting bodies. They differentiate, and up until 1898, they were actually classified as eukaryotes. Because what you saw were the fruiting bodies, and people thought that those fruiting bodies were actually part of the cellular slime molds. And if you look at them, they're about the same size. They look like cellular slime mold fruiting bodies. It wasn't until Roland Roland, who was at Harvard, he ran the herbarium at Harvard, germinated some of these things and realized, wait a minute, those things that are coming out of those fruiting bodies, they're not eukaryotic cells, they're bacterial cells. And so that was actually how the myxobacteria went from being classified as cellular slime molds into really realizing they were actually bacteria.
Wow, now I have a totally new vision of young Mitch Singer under the tutelage of his dad. You probably got a microscope or something when you work at.
Such a nerd.
That's
so
great, though.
Great.
That's a great vision for me to have.
Yeah. So that was how I got. Now, when I went to college, I actually wanted to be a vet. I took one animal science course. Thought it was the worst thing in the world. We spent 2 weeks on dog food labels, and completely turned me off. And then I went and washed dishes in Sam Kaplan's lab. And I eventually asked to work on a project, and he took me because I was the only one who didn't want to work on the hot molecular biology stuff. I wanted to work on lipids. So I did my undergraduate project on fat and did all kinds of things you are not supposed to do. Mouth pipetted chloroform, only passed out once.
Wow.
Spent my Saturday and Friday nights cleaning up P32 spills from, I ran the gambit.
Do you glow in the dark?
I have 2 children. Luckily, I was okay. The worst that ever happened was I had a P32 spill, and I had to disrobe in lab and walk around in lab stark naked except for a lab jacket until the postdoc I was working for could go to my apartment and get me a pair of clothes, which took him an awfully long time considering I was only about 3 blocks away.
Yeah, that was a little fun. So bringing this back, I just wanted to sort of ask you some questions about multicellularity, since you work on or had worked on stigmatella, and that's one of these examples that when I think of, a lot of biologists have this perception that only eukaryotes are multicellular. And so maybe we could start like really simply and just have you give us a definition of what you think multicellularity is or means.
Yeah. So in the bacterial world, at least, we view multicellularity as a process where multiple cell states can arise from a single cell state. And this dual role can occur simultaneously by some mechanism, either stochastic or not. And so you can look at a variety of bacteria and ask whether or not they form sort of the classic views of development. And we can start with the myxobacteria, because what happens is the myxobacteria, under vegetative conditions that's normally growing, they look like any other bacterial colony, but when starved, they do some incredible things that are relatively unique to that grouping. They have a series of behavioral patterns that you can see. They aggregate together to form these mounds. These mounds will then differentiate into spores. And not all cells make it. Some cells clearly have different pathways. Some cells die. And there is an auto mechanism for self-destruction. We used to call it, bacterial. Okay, wait, my brain's been turned off. Cell death in eukaryotes is called apoptosis. Cellular apoptosis, but it's not apoptosis because that's a very specific mechanism with P458 or whatever going on. But there is a series of programmed cell death that does occur. There are toxins that can be shown that are induced that kill the cell. It's believed that the cells then are cannibalized by those cells that are undergoing the rest of the developmental progress. So the colony survives genetically, even though individual members are killed in the process. The ratios of those cells that turn into spores can vary depending on how you starve the cells. And the end product is not a single cell type, but multiple cell types. We have these things called spores that are metabolically dormant. They can be dormant for hundreds of years. The spores that Thaxter looked at were fruiting bodies that he found in the woods. They weren't the cells of the vegetative state. And there's another form called peripheral rod cells, which are a form of cells that are not spores, but surround the fruiting body and are believed to potentially be a short to starvation. And we've shown in my lab using RNA-seq analysis that peripheral rods are a unique cell type compared to, say, vegetative cells, stationary phase cells, and spores, because they have a very distinct transcriptome, i.e. proteome, than other cells. And that's really interesting. So it's really a truly different cell type. So
if I were to restate this to you, so when I am teaching multicellularity, one of the things that I say is that a multicellular organism is one in which some cells in the body, whatever the body actually is defined as, give up the ability to do reproduction. But your definition is actually a little bit more precise in that you're saying that you have cells in the body, whatever that body is, having multiple potential developmental pathways and or outcomes. Is that fair?
And then functions, right?
And
so spores are long-term. And then, of course, we don't necessarily consider an outcome cell death, but that's a real function for the propagation of the species because the cells are dying. And you don't get any more terminal than dead. Right.
So
3-cell fates.
Right. So one of the bigger questions I always think of is that given that multicellularity in general has evolved multiple times independently, what would you think are the big advantages to multicellularity and just in general?
So that's a really cool question because it's true. Multicellularity arose many times, and it's arose several times in the bacterial world as well. And based on the eukaryotic definition, probably the best example of multicellular development in the bacterial world is filamentous cyanobacteria. Because in that case, cells become terminally differentiated, called heterocysts, where their only function is to make nitrogen, right? To absorb atmospheric nitrogen, stick it into an organic compound to make glutamine. That's their role, and the rest of the cells feed them, right? They feed them carbon, they provide the nitrogen. And that's actually the close thing of a truly eukaryotic version, right? And you can see this. You see the filamentous, and you see these spatially port-placed heterocysts. And I strongly suggest that you ask Professor Meeks, Jack, to come to talk to this, because he'll talk for hours. And cyanobacteria are really cool, because without cyanobacteria, we would not be here.
Right.
You know, I tell my BIS 2A students every time they walk by a puddle and see green slime, they should get on their hands and knees and thank that green slime, cyanobacteria, for producing the oxygen that allowed for the expansion of the eukaryotic world and multicellularity. And it's interesting, in the bacterial world, those that are mostly associated with multicellularity are those that are aerobic. And so this is a newer project I'm working on in my lab, and that is, what is the evolution of development in the myxobacteria? So if you look phylogenetically, myxobacteria are delta proteobacteria, and if you look at who are their closest relatives, who else are deltas, they are things like Geobacter and Delvibrio and Desulfovibrio. They are anaerobes, obligate anaerobes with small genomes. Okay. The myxobacteria are basically aerobes with large genomes. In other words, when they figured out how to use oxygen as a terminal electron acceptor, it opened up this incredible world. And all of these extra things in these large genomes aren't deltas, right? They come from other places. So they were very smart, evolutionarily speaking, in terms of hijacking and absorbing other genes that they could then exploit. So they rapidly grew out of this delta, this anaerobic type, and you can still see remnants of that. You can find myxobacteria that are taxonomically myxobacteria based on 16S that are obligate anaerobes, anaerobe myxobacteria. They don't develop. They have small genomes, and there's nothing too special about them except they're really unique myxobacteria. Then you can find myxobacteria that are aerobes and have small genomes. And then you find the aerobes with the huge genomes, almost as if there's, the stepping stones to become. Most myxobacteria are the larger genomes. They have some of the largest genomes in the microbial world, up to 15 megabases, which is bigger than haploid yeast. Okay, and where did they get these things? They did not reinvent cytochrome oxidase. They got it from somebody, and they tend to have gotten it from cyanobacteria. That seems to be the big play, and and some components from other proteobacteria, like some of the alphas and gammas.
Right.
So they clearly stole things. There's a lot of hanky-panky that goes on cross-species in the bacterial world, and they took advantage of that, and then they exploited that and became these multicellular issues. And the other thing, so why making spores? So myxobacteria, when we think of spores, we think of like mushrooms, right? That tend to want to have or trees with seeds. They want to spread that stuff out. In myxobacteria have to compete with other soil bacteria like Pseudomonads and Bacillus, who have doubling times of 30 minutes, 40 minutes. They can't compete. However, a colony of myxo can compete because they tend to prey on other bacteria. They digest macromolecules, and the best source of macromolecules are other bacteria.
So and that's interesting you bring that up, because if I am recalling correctly, I remember sort of this idea that maybe stigmatella functions almost like there's a group behavior,
the
or
wolf
group
pack.
dynamic, the wolf pack.
The myxobacteria in general have been talked about as being wolf pack predators, and they will actually swarm around prey and devour them. They secrete digestive enzymes that basically weaken their prey, and then they have toxins and interactions where they literally rub up against the prey, and they are degraded. And you can see beautiful movies that Hans Reichenbach made back in the 60s of myxobacteria eating Serratia, E. coli. And one of the nice things about working on myxobacteria is because it can be difficult to keep them nice and fresh when you grow them in a solution, because they don't do well, right? If you have a bacteria that can double every 30 minutes and they're doubling every 6 hours, the population gets overrun. But if you have a plate and it's contaminated, just wait a while and the mixos start coming up and they eat. You can see them eating other bacteria, and it's kind of cool. And we study this predation behavior in a variety of ways in my lab and in collaboration with others.
So if I would summarize, then advantages of multicellularity would be obviously some increased reproductive capacity,
right?
Maybe some of these more complex behaviors. I don't know if you would define that as
absolutely.
A
so I forgot to finish my story, but what Mixo does is Mixo doesn't make spores, so single spores can be blown away and start a colony, because a single spore has a very hard time competing. What Mixo does is it makes spores. It also makes slime that sticks those spores together, and you now have a ready-made colony. So the whole spore packet is kept together, and you have an instant microcolony when germination happens. So you don't have single spores making a population to be spread out. You have microcolonies spreading. So in a sense, the sporulation process isn't a dispersal mechanism so much as a retention mechanism of the colony. So now you have 10 to the 6 cells all stuck together, so that when you develop, or when you germinate, I should say, you have that microcolony there. And again, there's these beautiful movies of Hans Reichenbach with those little packets, especially you see in Stigmatella with those packets that come out. There's about 10 to the 5th cells in each of those packets. Those packets germinate, and you can see all these cells swarming out of that single packet. You've got your microcolony. Now your microcolony can now compete because it can secrete digestive enzymes, and Bacillus doesn't have a chance.
So I guess the last thing I would ask is, so if I understand correctly, Stigmatella spends the majority of its life as unicellular bacteria cruising around, and then they aggregate. And so,
they don't. That's not really true.
Okay.
So they don't spend their time as single cells.
Oh,
okay. I mean, single cells, but in a colony. They're very colonial. So individual cells, we think of bacteria as being like loners, right, out by themselves doing what they do. But the myxobacteria stay together. In fact, you can measure their growth rate, and their growth rate increases with the number of cells in the colony. Okay, and what that means is the more cells there are, the more digestive enzymes that can get out there, the more prey can be killed, and the more compounds that they can suck up.
So there must be lots of mechanisms for cell-to-cell communication.
There is. There's enormous mechanisms of cell-to-cell communication. There's a lot of cell-cell interaction. They have coordinated cell-cell movement. So the myxobacteria have 2 types of movement. They have individual movement called adventurous motility or amotility, and they have group motility called S-motility for social motility. And both are actually required for the survival of the colony. So you have these single cells that kind of go out to test the environment, and they come back in, and then these rafts of cells that go out to move the colony out in a specific direction towards the prey. So their behaviors are fascinating. I mean, if you look at it, it's almost like there's some form, I'm going to get in so much trouble if I say like an intelligence in that colony. I mean, it's not. It's all chemical interactions, right, that are driving these behaviors. But if you look at it, it's really cool.
Wow, it's like a whole other world. Well, thank you very much for coming on the show today, and definitely look forward to future discoveries from your lab. And thank you very much.
Oh, thanks for having me. It was great. Anytime.
Thanks for watching our video. If you enjoyed the content but would like an audio-only version, please go to iTunes and look up Tree of Life UC Davis. If you want to see more videos on YouTube, please click the subscribe button, and don't forget to hit the little bell icon if you want to receive notifications of new content.